Only 1 minute, please. Live stream is going on.
Perfect, sir.
Okay, you can start now.
Okay. Good morning, everybody. Today, I'm very happy that in our PhD webinar series, we are going to work with our seventh speaker today. Dr. Jasna Vijayan is the speaker today from Tamil Nadu. Hello, ma'am.
Hi, sir. Sorry, sir. I am from Kerala.
Oh,
My
okay.
university is in Tamil Nadu.
Okay, ma'am. Okay. Glad to know, ma'am. Glad to know. Very warm welcome to everybody. And now, in the interest of time, I would ask All India Microbiologist Society President Dr. Arvind Deshmukh to speak few words.
Hello. 1 minute, please. 1 minute.
Sure, sir.
Yes. I'm very happy today to inaugurate 7th talk of a PhD series. Actually, the main object of our PhD series is that we want to encourage young generation. The young generation is doing PhD, doing research, and we should be proud of young generation. So we started this series and we got very good response throughout the India. Today I am very happy to note that from Kerala, Dr. Jasna, she is going to deliver a lecture on distribution dynamics and molecular fingerprinting of viral communities. So it's a very, very interesting topic. We are recording this topic. It is available on the YouTube. This YouTube will be circulated throughout the India later on different groups. And these students will felicitate them by giving a memento and certificate from Microbiologist Society. And Jay sir is doing a very good job. He is organizing, he is taking effort. And I'm really thankful to Jay also for arranging this event. And thankful to Jasna ma'am for coming over on our platform and giving lecture. Thank you. Thank you.
Thank you very much, sir. I am glad that you are giving us this prestigious opportunity, and I am really glad to be part of the series, which is sharing knowledge. Hello, everybody. I would like to introduce our speaker, Dr. Jasna Vijayan. Dr. Jasna has done a PhD in microbiology from CSIR, National Institute of Oceanography, India. Ma'am has done her MSc from Bharathiar University, Tamil Nadu, and her BSc Microbiology from MG University, Tayam, Kerala. Ma'am has been associated as a project assistant at CSIR NIO for approximately more than 10 years. Ma'am is currently working as a UGC-Kothari postdoctoral research, which is actually a very prestigious scholarship in Dr. Mohammad Hatha's lab at Department of Marine Microbiology and Biochemistry, School of Marine Sciences, Cochin University. Ma'am has published more than 2 dozen international and national research papers, Ma'am has participated in various conferences and paper presentations, and ma'am has very prestigious record, and she is a renowned faculty, very experienced faculty. Hello, ma'am. You may please start your talk.
Yeah, thank you, sir. First of all, good morning to everyone. I'm very glad, thankful to the organizing committee, Arvind Deshmukh sir, and Jay for giving an opportunity to present this platform. So I'm just directly going to my presentation. Is it audible?
Yes, ma'am, loud and clear.
Yeah. So my PhD topic is distribution, dynamic and molecular fingerprinting of viral communities in Cochin estuary, India. I have done my PhD in CSIR National Institute of Oceanography, and I have registered in Varadhasan University, Tiruchirappalli, under the guidance of Dr. Parvati. She is a principal scientist in CSIR NIO. Now she moved to Cochin University as a professor. So she introduced me in this great field, that is viral communities, into my PhD topic. So I'm really grateful to her to giving this dynamic to complete this dynamic work. So I'm moving to my slides.
1 minute, Asna ma'am. 1 minute only. Hello.
Yeah, ma'am. Yes, sir.
I want to tell you, I'm very happy today. Renowned personalities of India are attending your lecture.
Thank you, sir.
Example, Dr. Prakash Halami, a very big scientist from CFTRI Mysore. Dr. Dayanand Akhzar, he is the Honorable Vice Chancellor of Gulbarga University. Dr. Swarnajit Singh. Hello.
Yes, sir.
Dr. Swarnajit Singh. He is a very big scientist from Chandigarh. So I welcome all them. Ajay Srivastav, he is also attending from Jharkhand. And Dr. Rachana, Dr. Anamika. So I'm very happy that renowned scientists of India have taken interest in your topic. Thank you.
Thank you, sir. So few years, that is now the world is suffering. Everyone is aware of the viruses because everyone is facing a big scenario now, this almost 2019 to this years. So everyone is familiar with the word viruses. So I'm just going through what is viruses. Everyone know, but giving a just introduction. The viruses are small microscopic particle which consist of either DNA or RNA surrounded by a protein coat. They have no metabolism and they are entering on the host organism and for their reproduction. So they are capable of infecting all life forms, that is from plants and animals to bacteria and archaea. So they need a host for their replication. Coming to marine viruses, like bacteria, we are much aware about the bacterial component, that is they are the normal flora and they are present in everywhere, but we are less aware of the marine viruses. They are highly abundant and ubiquitous, that is they are present in everywhere, near shore and offshore, tropical to polar, sea surface to sea bottom, and even in the ice, that is in Arctic and Antarctic ice, they are present. And the ocean water may contain about approximately 4 into 10 raised to 31 viruses, and which may transfer approximately 200 metric ton of carbon globally. And their abundance is varying in different aquatic systems. Actually, we are mainly focusing on the marine viruses because our institute is mainly Oceanographic Institute. And that's why I'm going giving an introduction about marine viruses. And in deep sea waters, their abundance is approximately 10 raised to 6 viruses per ml. And their abundance is varying in coastal waters. The productive coastal waters, they are in a range of 10 raised to 8 viruses per ml. And in sediment and soil, their number is comparatively higher. They are approximately 10 raised to 9 to 10 raised to 10 viruses per gram of sediment or soil. And in the marine environment, approximately 10 raised to 23 viral infections occur in every seconds. So this gives the significance that viruses are significant component in the marine ecosystem. Now, just we compare the relative biomass and abundance of prokaryote, protist, and viruses. Based on this pie diagram, you can see that biomass-wise, prokaryotes may comprises approximately 95%. But when we compare to their abundance, they are only less than 5% or 10%. But biomass-wise, viruses are very less, only 5%. But abundance-wise, they comprises 94%. So we have just go through their significance. So this much of viruses are present. Now we look into what are their significance. So they play significant role in microbial food web. We all know about the classical food webs, that is smaller organisms are eaten by the higher organs, that is phytoplankton. These are the base of the food chain, and they are eaten by the small grazers and small grazers to large grazers. Likewise, the energy is transferred to lower trophic level to higher trophic level. This is the classical food chain. And when they die or death occur, the decomposers such as bacteria act on it, and the energy is back into the system. But when viruses are present, viruses are capable of infecting all these life forms, such as bacteria, phytoplankton, zooplankton, everything. They are capable of infecting all this life form. So the energy, when they infect these life forms, they burst this host cells and liberating viruses into the system. So the energy, the energy is not transferred to higher trophic level. The energy is short circuiting in this small this is called microbial loop. So they play significant role in food web, and they play important role in nutrient cycling. In aquatic ecosystem, the nutrient cycles are significant. In nutrient cycle, what they are doing, bacteria, when they viruses, when they infecting this bacteria or phytoplankton, they lyse the cells. So when they lysing the lysed components such as cell debris, cell organelles, everything released into the water. So the cell wall components containing carbon components, the nucleic acid component containing nitrogen component, phosphorus component, and thereby they influencing these nutrient cycles. They play significant role in microbial diversity. How means they limit the population. If there this viruses are capable of infecting all the life forms, that is phytoplankton and bacteria, so they attack the this life forms, and thereby they limiting their population, and they play significant role in diversity. How means, you heard about the term transformation and transduction. That is when viruses infecting bacteria, the bacterial cell is bursting, and nucleic acid component, cell wall component liberating into the environment. So they may be utilized by other organisms. Or when the virus is infecting the bacteria or other organisms, they're taking some genes from the host organism, and when they infect other life forms, they are transferring to other life forms. So thereby they are influencing the microbial diversity. They influence the marine health by controlling the algal and bacterial population. That is, sometimes if enough nutrients entering into the system leads to some eutrophication. That is algal bloom or algal bloom like scenario. So when viruses are there, they have a more chance to interact with this bacteria or phytoplankton, and thereby controlling their eutrophication or not eutrophication, the bloom formation, and by the influencing the marine health. And this much of information are available in the many review articles and so this give me to that how many viruses are present in the aquatic system, are they active, what are they doing, how diverse they are. So for that, I have selected a study area as a Cochin estuary. Cochin estuary is one of the most productive estuarine system along the southwest coast of India. So I have studied viral distribution studies in Cochin estuary, viral processes in the Cochin estuary, their morphological and genetic diversity in the Cochin estuarine system. Coming to my work, I have selected this Cochin estuary because it is one of the most productive estuarine ecosystem in the southwest coast of India. They have a connection to the Arabian Sea. I have sampled in 13 stations along the Cochin estuary on a monthly basis. From 1 year's period, we sampled from this 13 stations. And I have labeled the stations at 1 to 13. Among these stations, 2 stations are barmouth, that is inlet station. Through this inlets, saline water, that is from the water, saline water from the Arabian Sea entering into the estuarine environment. And I have marked 2 stations here in the pink color. From these 2 stations, I have sampled on a daily basis for a 1 year period to estimate the viral and prokaryotic abundance from the Cochin estuary. For abundance estimation, I have collected the sample, fixed the samples using formalin, and filtered through the 0.02 μm pore size anodized filter paper, and stained with cyber green, and using an anti-fading solution also we used, and then count under the epifluorescent microscope. This is the epifluorescent image of and you can see that the large spots, which are viruses, and tiny spots are there. They are viruses. And based on my study, I have presented few pictures here. In our southwest region, we have 3 significant seasons, that is monsoon, pre-monsoon, and post-monsoon. That is June to September. June, July, August, September period we are considered. It's a monsoon period. Before and after period is a post-monsoon and pre-monsoon period. And in our estuarine region, we got viral abundance approximately in the 10⁻⁷ viruses per ml, and the abundance is comparatively higher in the pre-monsoon season, followed by post-monsoon and monsoon season. Along with the biological parameters, we analyze all the physicochemical parameters such as temperature, salinity, inorganic nutrients, everything. Based on that, the viral abundance is significantly influenced by we plotted a PCA analysis. Based on that, we got the most significant factor influencing the viral abundance is the prokaryotic abundance. Maybe it will be the host for viruses. That's why it is significantly associated. Moving to from the first observation studies, I have understand that this much of viruses are present in this ecosystem. So I have just looked into the what are the what role are they are doing in our estuary. For that, I have limited my study stations into 5, and this S1 and S3. These are the inlet stations. I already mentioned the previous slide. From these stations, the saline water entering into the estuary, and I have marked in green color. In our estuarine region, 6 major rivers are entering, which bring lot of fresh water to our estuarine system. And in the monsoon period, that is June, July, August, September period, the entire estuary behaves as a fresh water due to the monsoonal effect and due to the fresh water bring out by the riverine system. And in the monsoon period, just please observe this S1 and S3 station behaves as a mesohaline station. That is, this station only the salinity will be 15 to 18 ppt in the monsoon period. But all other stations behaves as a freshwater region. But in pre-monsoon period, that is, this S3 and S1 behaves as a euryhaline. That is, the salinity is higher. That is, the salinity is entering into the saline water entering into the system. And during the pre-monsoon period, the effect of fresh waters are less, and also the influence of rainwater also less. So these 2 stations behaves as a euryhaline region, and in between these 2 stations, that is S2 and S4, behaves as a mesohaline region, and the S5 station, it behaves as a freshwater in the pre-monsoon period. And this is the rainfall distribution pattern during my study period, that is June to September period. We got a higher rainfall, and compared to when we compared to the pre-monsoon period, we got lesser rains. And this is the ODV plot. That my study station S1 to SV plotted here. This S1 and S2 and S3 in the monsoon period, it is a mesohaline station in the monsoon period, This is the ODV plot. The color code indicates the salinity in all the stations, and the S5 station, it is purely the salinity is 0 to 5 range. The others the 3 stations behaves as a freshwater region, and S1 and S3 behaves as a mesohaline station. Whereas in the pre-monsoon period, the inlet station, that is the saltwater entering station, that behaves as a euryhaline region, and the S2 and S4 behaves as a mesohaline region, and S5 behaves as a freshwater region. And for this, we have collected sample on a hourly interval. That is, samples were collected from 2 seasons on an hourly basis, from morning 3 a.m. to next day next day 9 a.m. 3-hour intervals. We collected the samples, and the viral abundance in our study region ranges from 0.14 to 3.98 into 10⁻⁷ viral-like particles per ml. And we also estimated the viral production by modified dilution method. Based on that, in the monsoon period, we got higher viral production in the mesohaline station, that is in the S1 and S2 stations in the monsoon period, where in the pre-monsoon period, we got higher viral production in the S2 and S4 station. Coming to the viral mediated modality.
Hello. Hello,
Viral
Rasna
mediated
ma'am.
mortality. Yes, ma'am. Yes, sir.
Rasna ma'am.
Yes, sir.
It seems that there is a problem in sound.
Okay, sir.
Whether it is only for me or for others, I don't know.
Now I think she is audible.
Yes.
Okay, sir.
I think she is audible, sir. Yeah.
Okay, thank you.
Thank you.
So viral mediated mortality means the ratio of viral production and burst viruses. Hello, is it audible?
It's cracking in between. Can you please check your internet connection, ma'am?
Okay, sir. Is it okay now?
Yes, better. Please continue.
So the viral burst size is the number of progeny viruses released from a host cell, and the viral mediated mortality in our study region ranges from 0.3 to 51%, and it is also higher in the mesohaline region. And it was based on the percentage of bacterial production to carbon. That is, it ranges from 1.56 to 253 microgram per carbon per liter per day, and this is also higher in the mesohaline station. To confirming this cluster, this mesohaline and freshwater region, we go for a cluster NMDS analysis in the primer, and based on this analysis, we got monsoon, we got 2 clusters, and in the pre-monsoon, we got 3 clusters, that is mesohaline and freshwater region in the monsoon period, and whereas in pre-monsoon, we got 3 clusters, euryhaline, mesohaline, and freshwater region. And based on that, we conceptualized our study in a flow chart. That is, in the monsoon period, the mesohaline region, the viral lysis and viral activity is comparatively higher, and bacterial respiration and bacterial production also comparatively higher in the mesohaline station. And the studies were published in Fondius in Microbiology Journal. And next, I am moving to my another topic, that is, in the initial studies, I have studied the distribution pattern and what are the viral processes in the Cochin estuary. And now I'm moving to what are the morphotypes. What is the morphological diversity in the Cochin estuary? This is a TEM image. For TEM analysis, we collected the sample. We fixed the samples using formalin, glutaraldehyde, and we concentrated on the grid by ultra centrifugation method. And this mesh are filtered using filter, and we are observed under transmission electron microscopic analysis. And these are some pictures of viruses. You can see that this is the bacterial prokaryotic cell. Inside, the viruses are tightly packed. This is the starting stage of bursting. That is, when viruses infect a bacterial cell, it will lyse the cell and liberating viruses. This picture, the bursting is started This is completely burst prokaryotic cell, and we analyzed what are the four types of viruses present in the Cochin estuary on a seasonal basis. Based on that, we got in all our study region myoviruses. That is the prominent one in our study region, followed by SIFO and podoviruses. Myoviruses all are bacteriophages with significant variation in the morphology. And we also look into what are the prominent hosts for viruses. Based on that, we got short rods are more prominent to viral infection compared to other forms, and cocci are least susceptible to viral infection. And we also look into the average burst size of each bacterial cells, and based on that, from fat rods more viruses are produced. That is a burst size. We can estimate number of progeny viruses released from the host cell. And this study is published in PLOS One journal. And we studied the morphological diversity. Now we are confirming that by genetic analysis. For that, we collected the sample, a large volume of samples from the estuary. We limited our study into 4 stations because from each stations we are collecting large volume of samples, that is 200 liters of waters we are collected from each station. After collection, using tangential flow, this is the ultrafiltration system, we are concentrating the viruses present in the 200 liters to 300 ml, and from this 300 ml we are purifying the viruses, extracting the nucleic acid, and then we are going for sequencing. So when we get the concentrated, we should need at least 10 raised to 12 to 10 raised to 11 viruses. Then only we can go for extracting the nucleic acid, because we all know viruses are very tiny particles. So to get an enough concentration of DNA, we need more viruses. Then only we will get good sequencing results. So for sequencing, we are used linker amplified shotgun method for double-stranded DNA viruses. And based on sequencing analysis also, we confirm we got myoviridae is the prominent one, and followed by SIFO and podoviruses. And for PR4 station, this is a freshwater station. In these stations, most of the viruses are unknown, unclassified viruses. And based on this metavirome analysis, we got Synechococcus phages was the most dominant one in our study region, followed by Pelagibacter phage, Pseudomonas phage likewise. And we can also confirm that what are the phage type, that is Synechococcus phage SSM2 was prominent in this stations, whereas in station 2 it was Synechococcus phage SSKS1 is the prominent. Likewise, the strain also we can estimate from this station, from this study. And we also predicted what are the functional role of viruses in this estuarine system. We classified our functional annotations into 3 categories, that is molecular function, biological function, and cellular function. Major molecular functions are the ATPase activities, DNA binding activities, ATP binding activities, whereas biological functions such as DNA recombination, DNA replication, etc., and the cellular functions such as viral capsid, integral component membrane formation. These are the major functional diversities of viruses present in our estuarine system. And we also compare our metavirome globally. Based on that, we compare our samples from Scan Bay, Salton Sea, California Bay, the North Atlantic Ocean, Indian Ocean. Likewise, we compare our meta sequences. This is our samples, and we plotted a PCA. And based on that, our samples are more or less similar with Salton Sea and California and Scan Bay. And the study is published in Scientific Report Journal. And this is my PhD studies. I am very thankful to my host institute, that is CSIR National Institute of Oceanography, where I have done my PhD. And now I have working as a postdoc in Cochin University of Science and Technology. Thank you to all.
Thank you very much, ma'am. Thank you.
Okay, now is there any questions?
Anybody, you can unmute yourself and please ask questions if any.
Can I ask one question?
Yes, sir.
Did you come across bacteriophages?
Sir, it's not audible.
Did you come across bacteriophages?
Actually, we are not going for isolation studies. My colleague in my lab, they are doing on bacteriophages. Now my postdoctoral studies, I have proposed a phage therapy that is from aquaculture. I am trying to isolate phages. In my PhD studies, I didn't go for isolation studies.
Okay, but when you're isolating viruses from Cochin region,
Yeah,
you may also come across so many bacteriophages.
yeah, because
So
in
how
our
do you
study,
know? How do you know that you have a pure virus and you don't have any bacteriophage?
actually based on our morphological study and based on metagenomic analysis, we got myo, sypho, and podoviruses, and these are the major viruses which are infecting the prokaryotic forms.
I see. But you know, I'm sure along with the viruses, we got trillions of bacteriophages also.
Yeah, actually viruses mean it include bacteriophages, phytophages, everything it will include.
Yes, so I just want to ask you one simple question. How do you know that your sequence does not have bacteriophage sequence? It's a very interesting question I'm asking you, right?
Sir, it's not audible. Can you please repeat the question?
Yeah. I'm saying that how can you say that you got pure virus? There must be a lot of bacteriophages also.
Yes, sir.
So-
Surely it should be bacteriophages. We are
Yes,
not
yes.
isolating phages or anything based on the metavirome. That is, we are from the environmental sample, we are filtering more water sample and we are going for a nucleic acid study.
Okay. All right. So what is the best part of your research? What
Actually,
do you feel?
actually, we don't know much about viruses. In every system, they are playing significant role in every ecosystem. So we don't know much about viruses. Especially, I think this is the first study from Indian waters. My PhD work is study. This is the first study. We don't have any references. Before that, I think as a part of Tara Ocean expedition, the cruise group come across one of our stations from the Arabian Sea. Other than there is no study from Indian waters.
Very good. So you be the first to report that
From our
in
lab.
the
From
in
our
the
lab.
coastal, yeah, in the coastal water of Cochin, there is a microbiome. You know microbiome?
Yeah.
You know what is a microbiome?
It's the India microbial components.
Yeah, you have trillions of bacteria, you have trillions of fungi, and you
Yeah.
also have trillions of viruses
Yeah.
in the microbiome. Okay.
Yeah.
So you do one thing. If you do PDF, you plan a small study on microbiome,
Okay, sir.
and that is the current first
Right.
area of research.
Okay.
Okay.
Okay, sir. Thank you so much.
Thank you. Thank you.
Dr. Jasna, can you hear me?
Yes, sir.
Wonderful. First of all, I would like to congratulate you for taking such an extensive study in a totally new area, at least for the Indian microbiologist is concerned, where you studied the diversity of different viruses in the Cochin. More importantly, I would like to again congratulate you have carried out the research in our CSIR laboratory. I am from CFTRI Mysore.
Okay, sir.
You carried out this work in NIO Goa and
No, I carried out in NIO Kochi. Actually, this is the regional center.
Yeah, NIO Goa Regional Station Cochin.
Yeah.
I am aware about that. And as Dr. Saranjit Singh also belongs to our IMTech CSIR family, and he has pointed out What I can also say that this was truly a bioprospecting with reference to studying the different viruses and studying their diversity and getting three wonderful papers. So the PLOS one, then Frontiers, and then scientific report. Just somewhere I missed the link perhaps, that how you identify the host. Number one. Number two is again Dr. Swaranjit Singh also told that how you are sure that the sequencing which you are doing belongs to the only one type of virus. It can be the different type of virus because you are not enriching and making the library, right? But you are just whatever the pool is there, you are just concentrating that and then sequencing. So how you solve that problem?
Actually, the major problem we face is our study is even chem analysis, transmission and electron microscopic analysis. We don't have the facility, so we depend. We outsource the samples. That is for Dr. Pradeep Ram and Dr. Sime Encanto from CNRS France. They help us to get good images of this inside the bacterial cells, viruses. We can get with their help. We got this images and all. Based on morphological analysis, we confirm that.
So most of these are the bacteriophages only.
Yeah, and genetic analysis we used a linker amplified methods, and because we get only double stranded DNA viruses, and we annotated using different platform, NCBI, FGRAS, different platforms we used. Based on that, we confirm that it's a myoviruses and cifovirus. That is based on global comparison from this estuarine and coastal environment. The major one is myovirus. That is those infecting bacteria. That is a major dominant one in almost all ecosystem. We based on the review articles.
Any of the new viruses which you identified during the study?
Actually, from our lab, my colleague isolated some viruses from Marinobacter, and it is in the Google Scholar. You can get Marinobacter phage from Arabian Sea.
That was not reported before, right?
No. It's reported from the other countries, but in our region.
What are their biological role actually in the ecosystem or in the marine environment?
Yeah, marine environment. Because we need to know what are the diversities in our ecosystem, because we have a lot of different biologically different components in our region. Based on this analysis, by knowing this, we can isolate phages from the environmental, and we have different application. Like we can go for phage therapy like application, because in current days, antibiotic resistant and all is the major.
Yes, exactly. The first therapy is another emerging area. Just a part of what Dr. Swaranjit Singh will be interested in most of the seminar, asking about whether you are maintaining this viral stock or not, or how you are maintaining, or whether your guide is taking care of those viral stocks which you have studied.
Yeah, my guide is mainly focusing on this marine viruses along with bacteria, and now she's doing 2-3 kind of work. One of the major component is virus.
Now, are they stock of this virus?
Dr. Prakash, I will ask her one question.
Definitely.
Can viruses be lyophilized or freeze dried?
No, actually, we are making in a glycerol stock.
Okay.
We extracted from the sample where we are making in glycerol stock.
See, why I'm telling you, when Dr. Chandra Mohan was there, Chandra Mohan from NIO Goa, I came to NIO Kochi and I set up your lyophilizer.
Yeah, we have lyophilizer, but
that I set up and I trained the people there in NIO Kochi.
Okay, sir.
So I'm sure you can freeze dry viruses, and that is a very good method of preservation, like Dr. Prakash is saying.
Yeah, exactly. Fully appreciate, sir.
That's all I want to ask.
All right, so thank you very much. Dr. Vijayan, I would ask that you type your mail in chat box so everybody can connect with you if they have more questions.
Okay, sir. Thank you so much for giving this opportunity with privileged speakers. Thank you so much. Thank you so much, Deshmukh sir, for as a part of Microbiology Society of India to give it to present my PhD work. Thank you to all.
Thank you, ma'am. Thank you. We were happy that you presented this lecture. And thank you, everybody, for attending this on a Sunday. Have a nice weekend.
Thank you. Thank you, everyone. Thank you, Dr. Jasna, and congratulations once again.
Thank you, sir.